All right, assalamu alaikum everyone. I hope you're doing well. My name is Noor Waseem, and I'm really excited to be giving today's session, which is spermatogenesis and sperm defects. Personally speaking, I feel like this is one of the easiest lecture in the repro block, and I've enjoyed this block thoroughly throughout. And inshallah, it's going to be easy for you. I've tried my best to simplify the concepts for you guys. And inshallah, I hope my explanation really helps. If you have any sort of questions, feel free to text me, email me, or call me anytime. Inshallah, I'll be able to help you guys. Okay? So, spermatogenesis and sperm defects. Let's begin. So, before we dig in deeper to the topic, what is a sperm cell? Here is a sperm cell, and it is the cell of the male reproductive system that is responsible for fertilizing a female egg. Now, keep in mind that our sex cells, both in male and female, have 23 chromosomes, okay? So which means it is a haploid cell. So once it what do you call it comes in contact with an egg which also has 23 chromosomes, it fuses and it makes a cell that has 46 chromosomes, and as a result that cell will be in charge of becoming the embryo or the fetus in future. Okay, so keep this in mind that our sex cells are haploid. Okay, so what about the structure of a sperm cell? It has a head which contains the nucleus, DNA, and the acrosome. Acrosome has enzymes that helps the sperm to penetrate the egg. Then it has the midpiece, which is packed with mitochondria, which provides energy for movement. You know how mitochondria is the energy house of the cell? So this part gives energy to the cell in order to move around. Then we have the tail, also known as the flagellum. It helps the sperm swim to reach the egg. So general function of the sperm is like it travels to the female reproductive system, it swims using its tail, it penetrates the egg to combine their genetic material and start fertilization. A healthy sperm is usually small, fast, and it's built for survival to reach and fertilize an egg successfully. And we'll also be talking in the end about of cases of sperm that's not really healthy, right? And that leads to infertility. Okay, so this was a general introduction. Now, moving on to spermatogenesis. What is spermatogenesis? Now, it is a process of sperm cell development where spermatogonia, okay, or the stem cell, it divides and matures into spermatozoa or sperm cells. And this process happens within the seminiferous tubules, which is located in the testes. This overall process of spermatogenesis, keep in mind, it involves one mitotic division and two meiotic division. Okay, and in order to have these steps regulated, we have hormones like testosterone, FSH, and LH. Okay, we'll be talking about them in the upcoming slides. Okay, so moving on to this part of the slide, spermatogenesis is the process in which germ cells develop into spermatozoa. It has four main stages. As you can see, it's got like four main stages in order to convert into spermatozoa. This means like the special cells in the testis, which is the spermatogonia, they are gradually going to change into mature sperm cells, which can further go and fertilize an egg. Now, this process starts at puberty. Keep in mind that boys don't produce sperm until puberty, and this process happens within a protected area in the testis, which is called the blood testis barrier. Now, by the name itself, blood testis barrier, it's like it's keeping the sperm cells in the testis in order to prevent or like keep it safe from the outside blood. Okay, so for that reason, it is called the blood testis barrier, which keeps the developing sperms safe. Now, the hormones involved are FSH and testosterone. Very important point. Please keep this in mind. FSH, it helps the sperm to grow and develop. Okay, whereas testosterone, it is a male hormone that is going to help the sperm mature and keep the process going. Okay, please keep these two differences in mind. That FSH is good for growth and development, whereas testosterone, it's important for sperm maturation. Okay, now as spermatogenesis progresses, germ cells migrate from the basal to the adluminal region of the seminiferous tubule. Now let me show you something. Now this entire thing is the seminiferous tubule in the testis. Okay, the process of spermatogenesis happens from the basal layer, as you can see here, from the basal layer to the abdominal region. Or in other words, you can say they move from the outer part of the testis to the inner part where they're ready to become released. You see, they're getting released here. And the blood testis barrier protects them from harmful substances. Keep that in mind. Okay, now the entire process of spermatogenesis, it happens it takes approximately 72 days, and with new groups of spermatogonia entering every 16 days. So in simple words, making sperm, or this entire process, takes about 72 days, okay, from start to finish. But new sperm production begins every 16 days so that the body is now able to make fresh sperm. Okay, I hope that point is clear. So just so that no one gets confused between the days, so it's more like the complete process of sperm production, starting from spermatogonia to spermatozoa, it takes 72 days. Okay, every 16 day, new group of spermatogonia or stem cells come in, and they start their development. All right, so the high yield from this slide is, obviously you have to remember this, you have to remember the hormones and also this as well. Okay, and of course, you know, meiosis, it is a kind of division that introduces genetic variation. And as you see, like we have meiosis 1, genetic variation introduced, we have meiosis 2, further genetic variation introduced, and that is with the help of our cell division called meiosis. All right, I hope that's clear. Now, this is more like just a summary of everything I said. Like you guys can see, mitosis is spermatogonia to form primary spermatocytes. Meiosis, like I mentioned, 2 steps here and here. And then spermiogenesis is when spermatids, they change into spermatozoa, right? They've got to remove some of their contents in order to become smaller, have a midpiece, they grow out of flagella in order to form spermatozoa. Okay, and spermiation, another thing, is when sperms are released from the Sertoli cells into the seminiferous tubules. Now, if you could see this process of sperm cells being released is called spermiation. Okay, so they make their way further to the epididymis. All right, so moving on, let's talk about the chromosomal separation errors. Now, during meiosis, it's very important to have proper separation of chromosomes, okay? That is having homologous pairs in meiosis 1 or sister chromatids in meiosis 2. Now, the reason it is necessary is because with the help of meiosis, you need to preserve the genetic stability and maintain the correct number of chromosomes. You know how I mentioned that sperm cells or female egg cells, they have 23 chromosomes. It's really, really important to have 23 chromosomes or the correct number of haploid chromosomes in order to have a healthy cell. Okay, and to have the correct number of chromosomes, that's called euploidy. Okay, so again, just to summarize, chromosomes must separate properly to make sure that the new cell gets the right number of chromosomes. And if the sperm cell gets 23 chromosomes, the female egg cell has 23 chromosomes, there will be fusion that would result in a normal healthy individual having 46 pair of chromosomes. However, there could be cases when the sperm cells would divide and they would not have the correct chromosome number. Okay, and that is called non-disjunction. It means like when the chromosomes, they fail to separate correctly during the meiotic phases. Remember the chromosome separation, we're talking about the meiotic phases, not the mitotic. So that results in aneuploidy, as in not having the right number of chromosomes, and obviously the cell will be abnormal. Now, when these abnormal sperm or egg cells, they contribute to fertilization, embryo development, that can lead to miscarriage and sadly spontaneous abortion. So if an egg or sperm with the wrong number of chromosomes combines with a normal one, the embryo will not develop properly, obviously. And about 60% of miscarriages happen because of aneuploidy. Okay, and there are some people with aneuploidy who survive. Like I mentioned, 60% go to miscarriages or spontaneous abortion, whereas some of them who are able to survive do end up unfortunately having certain conditions called Down syndrome, which is trisomy 21, as in an extra chromosome 21, which causes intellectual disability and unique facial features. I'm sure you guys must have studied about it. Klinefelter syndrome, which is a male with an extra X chromosome, and that leads to fertility issues and other physical traits in the male. Okay, the third one that we have is Turner syndrome, where there is a missing one X chromosome. You know how female has XX chromosome and male has XY? In this case, in Turner syndrome, the female has only X chromosome, like the other X chromosome is not there, which will obviously affect the growth and reproduction. All right, so please keep this in mind. These could be high yield. All right, now, although this picture looks really dense, trust me, it's not. I'll definitely simplify it for you guys. Okay, now, you know how we mentioned that the process of spermatogenesis happens within the seminiferous tubules? Okay, so keep in mind that this entire part is a seminiferous tubule. Okay, the seminiferous tubules have certain cells which are called the Sertoli cells. And right outside the seminiferous tubule, we have another kind of cell, which is called the Leydig cell. Okay, now, this diagram, it overall illustrates the hormonal regulation and the cellular processes of spermatogenesis within the seminiferous tubules, like I mentioned, of the testes. And something that's really important here is to ensure proper growth and development, or to ensure proper the process of spermatogenesis, the hypothalamic pituitary gland axis plays a really, really, really important role. A single hormone not working here is going to mess up the entire process, right? So what happens is that our hypothalamus, it releases a hormone which is called the GnRH, gonadotropin releasing hormone. Now, that GnRH acts on the anterior pituitary. What happens? Now, anterior pituitary is going to release LH and FSH, the luteinizing hormone and the follicle stimulating hormone. LH, keep in mind, LH hormone, it goes and binds with the Leydig cell to produce testosterone. Now, a way how I remember is that LH goes to Leydig cell is because L for LH and L for Leydig cell. Okay, mnemonic to remember this. So LH, it stimulates the Leydig cell to produce testosterone, right? So we've got one hormone, two hormone, and when LH stimulates the Leydig cell, we've got a third hormone which is called testosterone. And if you guys remember, in my previous slide, I mentioned that testosterone is responsible for the maturation of sperm, right? Maturation as in the growth, not really the growth, like, you know how spermatogonia to spermatocyte, and then when spermatids, they need to be further get matured into spermatozoa. Now here, the one hormone that plays a very vital role is testosterone. Okay, we have spoken about LH. FSH, it acts on Sertoli cells here. You see FSH going here. It acts on the Sertoli cell, and it promotes spermatogenesis and release of another substance which is called the androgen binding protein to maintain high local testosterone. So somehow FSH goes to Sertoli cells, produces the substance called androgen binding protein that in return allows more testosterone to be produced. So as you see, like, this entire system is really linked with each other, right? So it's really important to have a well-developed HPG axis because it plays a really crucial role in this process. So LH goes to Leydig cell, produce testosterone. FSH acts on Sertoli cells, promotes spermatogenesis and the release of androgen binding protein in order to maintain high levels of testosterone. Okay. Now, Sertoli cells, right? They, like I mentioned, they help produce androgen binding protein. Another thing that Sertoli cell produces is inhibin B. Now, this inhibin B, it is very important as it provides negative feedback to the pituitary to regulate FSH levels, right? So if the Sertoli cell feels that, okay, I've got enough FSH, I don't need any more, it's going to secrete inhibin B, and inhibin B is going to go back to the pituitary and inhibit the production of FSH for some time, okay? So here it plays a very important role in negative feedback. Okay, other than that, like, the process that's happening here is like We have got spermatogonia. They undergo mitosis. The results, the primary spermatocyte is gonna undergo meiosis, obviously reducing the number of chromosome by half. And then spermatids are developed. And then spermatids, they are further matured through the process of spermiogenesis, and they develop into spermatozoa. They gain tail for movement, and this is the overall process, okay? And the mature spermatozoa, as you can see here, the mature spermatozoa, it is released into the lumen of seminiferous tubule, and it is eventually transported for ejaculation. All right, I know it's a lot of information. I'm going to quickly summarize it for you guys. So this entire image shows spermatogenesis. This is the seminiferous tubule. The seminiferous tubules has Sertoli cells. Right outside, or like you could say close by to the seminiferous tubules, we have the Leydig cell. Okay, now the process starts with spermatogonia, which is the stem cell, and it divides and changes step by step into mature sperm cell, as you can see here. Hypothalamus releases GnRH, which tells the pituitary gland, hey, can you please produce LH and FSH? LH and FSH is being produced. LH, axon Leydig cell, produces testosterone, important for sperm maturation. FSH, it acts on the Sertoli cell, which supports and nourishes the growing sperm. Okay, and Sertoli cells also produce another substance which is called inhibin B, which is important for negative feedback. All right, so inside the tubules, like cells, they move from the outer edge to the center, gradually maturing into the spermatozoa, and eventually being released into the lumen of seminiferous tubules to get ready for ejaculation. All right, I hope it's clear. Does anyone have any question? All right, I think I can go ahead. Now, steps of spermatogenesis, honestly, nothing different. It's literally the same thing. It's just that we're going to break it down into like 3 to 4 steps, and it's pretty simple to remember, okay? You could say it's a revision of what I've just told you guys. All right. Now, step 1 is mitosis of spermatogonia. You know how we mentioned that the process of spermatogenesis starts from the basal compartment to the adluminal compartment. So it goes from the outer end to the center. So the first step 1 is basically, remember this, mitosis of spermatogonia. And we know when spermatogonia undergo mitosis, they produce something which is called primary spermatocyte. Khalas, that is it. What do you guys need to remember for step 1? Okay, and like you guys can read it you guys can read the lower part later on. And now let's move to step 2. Just give me a minute. Okay, I think my Apple Pencil have stopped working. All right, no worries. I can explain you guys without my Apple Pencil. Okay, so this was step 1. The process starts with spermatogonia, stem cell, which divides to keep a supply of cells for sperm production. Now, some of these cells would become primary spermatocytes. Like I mentioned, step 1 is mitosis of spermatogonia into primary spermatocytes, which will later go through meiosis to become sperm in later steps, right? So step 1, again, mitosis of spermatogonia to primary spermatocyte. That's it. Step 2 is when primary spermatocytes is being converted to spermatids, okay? Now, the primary spermatocytes, remember I mentioned it goes to meiotic division, meiosis 1 and meiosis 2. So that is basically step 2. Both meiosis, converting primary spermatocytes into spermatids. Now, step 3 is spermiogenesis. Spermiogenesis is when the spermatids mature into spermatozoa. Let me take you guys to the picture up there. Here, the last one. I wish my Apple Pencil was breaking. So it's basically when the spermatids are being converted into spermatozoa. Okay, that's the process of spermiogenesis, right? We're done with 3 steps, pretty simple. And to convert spermatids to spermatozoa, there are a couple of things that take place, like you guys can read it, which is basically developing a head, a midpiece, a tail for movement, having the production of acrosome, removal of extra cytoplasm. Because if you guys see here, the spermatids is actually a big cell, right? So it gotta cut down its cytoplasm in order to get converted to a spermatozoa. That's the process of spermiogenesis. All right. Step 1 done. Step 2 pretty simple. Step 3 done. So that's it. This is basically a summary. And if you guys want, you guys can go through it. And step 4 is spermiation. And spermiation honestly just means the release of sperm cells from the Sertoli cells into the lumen of seminiferous tubules, right? It's literally the release of cells. Spermiation, the release of healthy sex cells. Okay, so spermiogenesis, like I mentioned, the conversion of spermatid to sperm. You guys can read it. It's pretty simple. Like what's written here is that it forms a head which contains the sperm's nucleus, acrosome. It has a very important enzyme called hyaluronidase and acrosin, and that the reason why it's important is because it helps to break down the barrier around the egg cell. So once the sperm cell is healthy and it has acrosomal enzymes, it's going to be very easy for it to penetrate the egg and start fertilization. Okay, and the tail is responsible for sperm movement, enabling its motility. Obviously, the flagella. And midpiece, it has the mitochondria, and mitochondria is a powerhouse of the cell, right? Gives energy for the cell. Now, this was what Dr. Abdul Jabbar added, and like it's pretty simple. Honestly, it's self-explanatory. Spermiogenesis and spermiation. Spermiation, I just explained to you guys that it's just literally the release of spermatids into the lumen of the seminiferous tubules, whereas spermiogenesis is the conversion of spermatids to mature spermatozoa. Keep in mind that spermiation happens after spermiogenesis. All right, and you guys can read it. Yeah, pretty simple. And up till now, I feel like everything was really easy to understand. And moving on to these, the function of Sertoli cells, a little bit complex, but honestly, like if you understand, it's very simple. Okay. Now you remember how I mentioned the seminiferous tubules? They have Sertoli cells. Oh, I'm sorry about the spellings. It's not S, it's C. Sertoli cells. So Sertoli cells have 3 functions. One is supportive, one is exocrine, and one is endocrine. Now, like we all know, the basic definition of exocrine is that it refers to the secretion of substances into ducts or external environment. Okay. Now, in this context, the Sertoli cells, they produce and release the fluid that helps transport sperm from the seminiferous tubules to the epididymis. Okay. Endocrine, on the other hand, it refers to the release of hormones into the bloodstream to regulate these physiological functions. And Sertoli cells, they have got receptors for hormones like FSH, a follicle stimulating hormone, and we'll discuss about it, like the receptors it's gotten on it. All right. Okay. So talking about the supportive or the nursing function, you guys can read it. I'm just going to explain it. So Sertoli cells, they form and break junctions between the developing sperm cells, allowing them to mature. Now, they also help to maintain the blood-testis barrier, which protects the developing sperm from harmful substances. Remember that Sertoli cells, they remove the excess cytoplasm from sperm cells through the process of phagocytosis. All right, which is cellular cleanup. Remember how I said that spermatids are converted to spermatozoa, and in that process, in the process of spermiogenesis, the cytoplasm need to be removed, right? So Sertoli cells do play a role there by removing excess cytoplasm. Sertoli cells, under the supportive functions, they are also responsible for transporting nutrients and signaling molecules from the blood to the developing sperm. Okay, because remember I said that there's a blood-testis barrier, and the reason we're having this barrier is because we want to keep our sperm cells safe within the testis, but we like safe from the outside blood. However, we do need nutrients, right, and oxygen from the outside blood. So as a result, Sertoli cells play a role here by getting those nutrients for the developing sperm. Okay, and they also release something which is called the paracrine factors. Like they're basically chemicals, cell-to-cell chemicals that regulate sperm maturation. So just remember a couple of points here is that it preserves the blood testis barrier. It removes excess cytoplasm through phagocytosis. It transports essential nutrients to help support the sperm growth, and it also releases paracrine factors that help regulate sperm maturation. Okay, we're done with supportive functions. Exocrine functions. Now, in exocrine functions, the Sertoli cells, they produce a kind of fluid that helps move the immature sperm from the seminiferous tubules to the epididymis. Okay, sorry, not immature, immobile sperm, right? So when a sperm is immobile, when it gets fluid, obviously it's going to move, right? So it helps make the sperm mobile. Right. So Sertoli cells, another function it has under exocrine is that they produce the androgen binding protein, like I mentioned above, if you remember, which regulate the testosterone level in the testes. Remember, testosterone is really important for maturation of sperm. Also, they control when sperm cells are released into the lumen. So basically, Sertoli cells also help control the process of spermiation, the release of cells. Okay. Endocrine, now Sertoli cells, they express receptors for testosterone and FSH, which are key hormones for sperm production. They synthesize another hormone, you could say, which is anti-Mullerian hormone, AMH, and that is released during fetal development, which prevents the formation of female reproductive organs or structures. So AMH, or the anti-Mullerian hormone, the moment it's produced, it means that the baby is a boy, okay? And if it's not produced, the baby is a female, because the baby is eventually gonna have female reproductive structures, right? So because for example, if you're having a male baby, Sertoli cells are gonna help with the synthesis of anti-Mullerian hormone, and with that, the baby is gonna have male reproductive structures, and the baby is going to be a boy. Okay, and they also help aromatize testosterone into estradiol, which is influencing the local testicular function. Again, the role of testosterone is really important because it helps in sperm maturation. Okay, and another endocrine function it's got is the synthesis of inhibin, like I mentioned above, inhibin B, which is a hormone that regulates FSH secretion, right? It's basically a hormone that helps in negative feedback. All right. Pretty simple. Just remember the points that are highlighted in pink. It's you could be asked in your exam, what are the exocrine functions, what are the endocrine functions, and honestly, if you have a good idea of this image that I've showed here and the explanation I've given you, it'll be very easy for you to remember the supportive exocrine and endocrine function. All right. So moving on to the next slide, which talks about decapacitation and capacitation. Now, before I come to decapacitation, let me explain you what capacitation is. Capacitation is a process that the sperm should undergo in the female reproductive tract to become capable of fertilizing an egg. Okay, so the process of capacitation happens in the female reproductive tract in order to fertilize an egg. So how does that happen? It basically involves changes in the sperm membrane, and it removes certain proteins from the sperm. Now that removal of certain protein is going to allow the sperm to penetrate the egg, right? So capacitation happens in the female reproductive tract. The main aim of capacitation is to allow the sperm and egg to fuse, is to make the sperm capable enough to fertilize an egg. That's capacitation. So what's decapacitation? Obviously, the opposite of capacitation, where the sperm is kept inactive, or the sperm remains inactive or is unable to fertilize an egg. And it is regulated by specific proteins, which is present in the seminal fluid, which prevents the premature activation before reaching the female reproductive tract. Okay, so you could say that when the sperm is still within the male reproductive organs, it is decapacitated, and when it enters the female reproductive organs, it's gonna undergo the process of capacitation, which will make the sperm capable enough to fertilize an egg. Okay, and why is capacitation and decapacitation important? Now, first talking about capacitation, it is very essential to undergo capacitation if there should be fertilization, right? So without capacitation, the sperm cannot even penetrate the egg in order to fertilize, right? So, as a result, no embryo formation. And it also, the process of capacitation, it prepares the sperm for acrosome reaction. Remember I said acrosome has enzymes, and this reaction helps sperm break down the outer layer of the egg, which is the zona pellucida, and it also enhances sperm motility. All right. And why is decapacitation important? So it's the opposite of capacitation. And decapacitation, like I said, that it prevents premature activation. So it keeps the sperm inactive in the male reproductive system and seminal fluid until they reach the female reproductive tract. Also, it ensures proper timing, as in it prevents the sperm from wasting energy and undergoing capacitation too early, ensuring they're functional at the right place and at the right time, which is within the female reproductive tract. And that is when capacitation starts happening. Okay, so now talking about the information here, I'll be explaining it. You guys can read it. So decapacitation, like I mentioned, the opposite of capacitation. Here, the sperm released from the testes, it cannot fertilize the egg immediately, right? The reason is because they carry certain proteins that prevent activation, and they're maintained by interactions between cholesterol, phospholipids, and fibronectin-like substances. Now, this protein is not going to let the sperm waste energy, okay? It's only going to be removed when it enters the female reproductive tract and the sperm has started to be capacitated. Now, decapacitation, it also prevents the premature activation of sperms, like I just mentioned, before they reach the female reproductive tract, okay? And capacitation. The process of capacitation takes around 4 to 6 hours in the female reproductive tract before they are able to fertilize the egg or before they're able to reach the egg. So sperm must undergo capacitation in the female reproductive tract, and this process happens in the oviduct or the fallopian tube, and involves the biochemical changes to make sperm functionally mature. And there are key changes in the in the process of capacitation, which is basically the shedding of extra cholesterol. Remember, like we mentioned here, there were certain proteins that were kept with the interaction between cholesterol, phospholipid, and fibronectin. The moment they start to begin being capacitated, they start shedding that extra cholesterol, which makes their membranes more flexible. They lose certain proteins that were blocking them from binding the egg. What happens is calcium enters in. It helps the sperm release the enzyme or the acrosomal enzyme to penetrate the egg, and their movement and energy increases, right? So that makes them mobile, that makes them fast, and that makes them ready to be fertilized. All right. And this is decapacitation and capacitation. I really hope I've simplified the concept for you guys. Then now moving on to an important topic. We're almost nearing the end, which is infertility and sperm defects. All right. If you guys remember, I mentioned about the healthy sperm in the beginning, how it's supposed to be like, how its tail is supposed to be like, one tail, a proper headpiece, having an acrosome, having a midpiece, which has mitochondria, which is the powerhouse of the cell. However, unfortunately, in some people, there could be defects in the sperm, and that could lead to male infertility. Okay, so let's talk about it. So the changes in these is basically abnormal changes. So there could be sperm morphology defects that can lead to functional issues, or there could be genetic abnormalities. Now, when I mean sperm morphology defects, it means the problem within the sperm cell itself. Maybe something is wrong with the nucleus. Maybe the sperm has more than one tail. Maybe the acrosome doesn't have enough enzymes. Maybe there's something wrong with the midpiece, right? So it's like every piece works together in order to form or in order to have a healthy sperm cell, right? So it's a very interconnected system. So talking about sperm morphology defects, as in problems within the sperm, there could be chromatin condensation abnormality, which is going to affect the DNA integrity, because the the chromosome might not be condensed well enough, right? So that's going to affect the integrity of DNA. There could be acrosomal reaction defects, which is going to prevent successful penetration of the zona pellucida. Remember, acrosome, once it enters the female reproductive tract and starts going near to the egg, it's going to release its acrosomal enzyme that is gonna help neutralize the layer around the egg, which is a zona pellucida, and as a result, the sperm cell can enter in. Okay, there could be motility issues caused by tail abnormalities that hinder sperm movement, like for example, more than one tail, uh, or sometimes the tail isn't even there, or sometimes the tail is there but it does not move enough, right? So it cannot swim across. And also increase apoptosis or necrosis, which is reducing the viable sperm count. Okay, spoken about sperm morphology. Now, moving on to genetic abnormalities, which is the problem with which is a problem within the genes or chromosomes. Okay, and that's gonna lead to, like having different morphology of the sperm egg, and that's gonna have other problems. So let's discuss what exactly genetic abnormalities result into. There could be globozoospermia, which means that the sperm head is round. It's really round and it does not have acrosome. This is a picture of a sperm that is globozoospermia, round-headed sperm lacking in acrosome. There could be sperm macrocephaly syndrome, which means where the sperm heads are abnormally large, right? It's different from globozoospermia. Please keep that in mind. There could be multiple tail abnormalities, like having one tail, two tail, three. Some of sperms have more than two tails, which is obviously leading to impaired motility. Headless spermatozoa, obviously, it's going to lack the genetic material because the head of the sperm has nucleus and the nucleus has chromosomes, and chromosomes play a very, very vital role after fusion and development of a zygote, right? So if the sperm has no head, so it's going to lack the functional genetic material for fertilization, right? So again, abnormal sperm. All right, so you guys can see here, you can see we have got a normal sperm, a condensed acrosome. You see there's a lot of acrosome in the second sperm here, a lot of acrosome enzyme. Some of them have got like small head, some of them have got like really large head here, double-headed, double-tailed, and an abnormal midpiece, right? So either of this abnormality, although the sperm, you might think that, okay, although like the head might be big, there might be more than one tail, the midpiece is big, it actually has everything, but unfortunately, due to different morphology or some genetic problems, that's going to lead to infertility. All right. And globospermia, it's also, you could also think of it as balloon on a string. That's going to result in defective fertilization. All right. Now, moving on to more abnormal changes is, well, first one, delayed or reduced acrosomal reaction. So sperm with structural abnormalities, they might have difficulty undergoing the undergoing the acrosomal reaction, as in the release of acrosomal enzymes is going to be difficult, which is essential for penetrating the egg's protective barrier, which is a zona pellucida. This can reduce the success of fertilization. All right, because you need to have enough acrosomal enzymes in order to neutralize a part of zona pellucida to enter the egg. If you don't have enough acrosomal enzymes, that's not going to trigger the acrosomal reaction, and as a result, this would reduce the chances of fertilization. Now, effect of temperature, really important. So certain febrile illnesses, like chickenpox or pneumonia, they can temporarily affect the sperm's quality, because a fever, it can lead to decreased sperm motility and abnormal sperm shapes. But these effects typically recover within 4 weeks or when the fever is over, because increased temperature, keep in mind, that's going to decrease sperm motility. And if a fever occurs during spermatogenesis, which is the final stage of sperm maturation, remember, the conversion, not the conversion actually, it was actually the conversion of spermatids to spermatozoa. If the fever happens during step 3, like step 3 of spermatogenesis, the percentage of normal sperm may decrease by 7.4%. Keep the percentage in mind. Another abnormal change could be for the sperm could be as a result of testicular cancer, because it can lead to an increase in sperm abnormalities, which is microcephalic spermatozoa, sperm with small heads, defects in the post-acrosomal region. That's going to affect fertilization again, because the acrosomal reaction or the acrosomal enzymes are not being released or triggered. Acrosomal abnormalities, which is hindering the egg penetration, uh, excess cytoplasm and short tails, both of which will obviously impair sperm function and motility. All right. So please also keep this slide in mind, because you could get questions from this part. All right. And yeah, one more thing that I forgot to mention here is varicocele. Oh my God, I think I forgot to add the picture for varicocele. Anyways, I'll show you guys the picture. So what is varicocele? It is a condition that is characterized by the enlargement of veins within the scrotum surrounding the epididymis and testis. You know what? Let me show you a picture actually. Varicocele. I think Dr. posted. Yes, here is what varicocele. The one on the left is normal, and the one on the right is abnormal, and that's varicocele. You know, enlargement of veins within the scrotum surrounding the epididymis and testis. Now, what will this lead to? It leads to increased scrotal temperature. And remember, I mentioned that increase in temperature is going to impair spermatogenesis or sperm production, and even the mobility of sperm. So varicocele, it is linked to poor sperm quality, including defective motility and higher incidence of coiled sperm. Like you could have coiled sperm tails here, which could affect the sperm's ability to swim efficiently. Also, it is unfortunately one of the most common causes of male infertility, affecting around 10 to 15% of men globally. All right, so something to remember here is varicocele. Please remember, it's really important. And finally, our last slide here, although it's like all pictures, but let me give you a small overview of what exactly this picture is trying to say. So we see the female reproductive system here. We see the sperms entering through the cervix, vagina, and they're headed through the fallopian tube or oviduct towards the egg. Okay, now how do sperms travel all the way along and are attracted towards the egg? That's the question here. And the answer is through the process of chemotaxis. All right, so sperm concentration and motility, keep in mind concentration and motility, they're important in order to have successful fertilization. Now, here, the role of chemotaxis plays a very vital role, which is to help or guide the sperm move towards the egg. You see, like we've got like countless sperms here, but only one sperm is going to go and fertilize the egg. And how is that one sperm going towards the egg? That's only when chemical signals are released by the egg, and that signals released by the egg, they're going to attract the sperm. They're going to attract the sperm, which is going to help them navigate the female reproductive tract. So, when the moment sperm enters, right, the egg is going to release certain molecules. That's called chemotaxis. The process of chemotaxis is going to attract these sperm cells all the way towards the egg. So that's chemotaxis is basically the process of attraction between the sperm and egg. And that's also, like it helps them navigate the female reproductive tract. Additionally, there is another process called thermotaxis. That is temperature gradients within the female reproductive system. It assists in directing the sperm to the site of fertilization. You see the temperature over here is 34.7, the temperature over here is 36.3. And the temperature gradients, they assist or help the sperm to move from one side or one compartment to the next compartment of oviduct or the fallopian tube. Also, these mechanisms, they ensure that the most motile and the most functional, or you could say, like, in other words, the healthy sperm or the competent sperm reaches the egg, and that increases the chances of conception. That's it. That is what the slide is about. So, like, just to simplify again, overall, like sperm, it needs to swim through the female reproductive tract in order to reach the egg. The egg releases chemical signal that guides the sperm in the right direction. That process is called chemotaxis. Additionally, slight temperature differences in the female reproductive tract helps the sperm move towards the egg. That is because of thermotaxis, temperature gradients, and these natural signals, which is thermotaxis and chemotaxis, it help increase the chance of fertilization by ensuring that the healthiest sperm or the most competent sperm reaches the egg first. That is going to result in the process of fertilization, development of zygote, embryo, fetus, and eventually a healthy baby. All right, and that's pretty much what our topic was about. And if you guys have any sort of question, please feel free to ask me. Please keep in mind the high yield details that I've mentioned to you guys. If you remember that, inshallah, you're gonna ace questions from this topic. Also, just a little bit of tip is that the reason how I approached every lecture in Endo Repro was that I did the topic, and soon after the topic, I used to do questions from that topic only. Like, for example, if I did this lecture and I'm confident with my studying, I would go back to my past exam questions, or I would open previous batch exam committee questions, go to the specific lecture, and do the questions. This would like help me preserve important concepts, and like, you know, I would gain confidence doing questions after every lecture, because like it made me feel that yeah, I know I'm able to attempt questions. So that really helped. And yeah, that's it. If you guys have any question, please feel free to reach out to me through my phone number, through my email. I will be more than happy to help you guys. And if you'd like to give me feedback, feel free to scan the QR code and yeah, drop in your feedback. And thank you so much, everyone. Thank you so much to the host. I think his name is Omar. Thank you so much, Omar, for recording this session. And inshallah, I hope this is really helpful to you guys. All right. Thank you so much.